Could somebody confirm you can hear me?
I can hear you.
Great. Thank you also. Let me close that down. Let me see. Ah, I did. All right. Let me share my screen here. I can figure out how to do this. I got too many Zoom windows open because it's still making the file for the lecture. There we go. All right, you should be seeing my screen now. I have made it so that the Zoom meeting, we now have a link over on the left. And so if you click on it, I haven't ever done that yet, so I don't know what it looks like, but let's come on over here. It doesn't have anything here, but if you look at the all my Zoom meetings recordings, hopefully you'll get a link for the class, and then you can just click on, you probably want to have start, but I don't know what you're supposed to say. It might be join. And of course, the other way is to get in as you've been doing, and that works too. Um, let's close that down. Close that down. Let me state that I have graded all of last week's assignments. And that would be the worksheet for the lecture. Where is that? And that's worth 2.5 points. And then the worksheet for lab 00, also worth 2.5 points. And worksheet for lab 01, also worth 2.5 points. Normally we have the labs worth 5 points, but the first two are only worth 2.5 points. If you did not get 100% on the lab worksheet and the labs only, you may resubmit it by this Saturday for up to full credit. You have to have correct answers, but if you have correct answers, uh, you will get a full credit. And I've told you where the problems are on the lab. I mean, which questions were answered wrong. So that's the way I work the lab. It's a learning experience, and you will get it graded 2 times. The first time, if you don't get 100%, you can always do it again later to get 100%. Any questions about any of that? I have not talked, I've not graded any of the extra credit assignments. Um, the extra credit picture isn't even due this weekend or last weekend, but the worksheet was. I haven't graded that. Not that worksheet, sorry, the extra credit study sheet. I think that's about it. Any questions on anything? If not, let's go to worksheet lab 2, where we'll talk about lab module 2, aseptic technique, and talk about worksheet for lab 2. And let me see if there's something else I need to talk about. Yeah, you need to talk about we need to talk about the infectious disease research project. So let me go ahead and discuss that first, and then we'll talk about lab 2. So the infectious disease research project is where you're going to be picking a disease. It has to be an infectious disease, and write a paper on it. Let me see if I have that someplace else. There it is. think that's the latest one. Got too many there. That's the trouble. So this is a 100-point lab project where you pick an infectious disease and then clear it with me. Everybody will have a different infectious disease while you're clearing it with me. And then I want to make sure it's an infectious disease as well as trim it to manageable portions. Like some people want to do something like pneumonia, and I'll tell you no, because there's too many different agents which can cause pneumonia. If you want to do pneumonia, you have to pick one infectious disease which causes pneumonia and then write your report on that. Is that clear? So everyone will become an expert on one infectious disease. And unfortunately, you will all be taking different infectious diseases. The good news is because you're all taking different diseases, I won't be quizzing you on it. All right, you can read about the rationale, the project format. Pick an infectious disease, check with your instructor about that disease. As long as you're the first person who asked for that disease, you'll get it. If you're the second person or some later person to ask for that disease, you will not get it. There are some diseases which are very popular, like tuberculosis, and the first person who asks for it will get it. I should try and figure out how to split that one up, but I can't think of a way to split that one up. For HIV and AIDS, I can divide it into HIV-1 and HIV-2. So there are some tricks you can do. And I will do that, like with the flu, I'll divide it into influenza A and influenza B and influenza C. Then you're going to perform a search of the literature, use books, magazine, news media, scientific journals, the internet. If you use the internet as well as certain magazines, make sure they're reputable sources. A reputable source will be one that is unbiased, and generally, they should be reviewed sources. So if you're getting something from, I don't know if you guys even know Rush Limbaugh, but somebody once gave me a report that is all of his references were something like Rush Limbaugh and the Republican Party and maybe some from the Baptist church down the street, something like that. And I said, you know, these references aren't good references. You want a reference that is unbiased and will give you both sides of the, I don't know, both sides of the concept, both views of the concept. And when you have a source which is biased because they're promoting one side or another, it's not a good reference. And so you must have good references. All scientific books you'll see are reviewed by other authors, and then all scientific papers that are published are reviewed by other scientists, usually two other scientists. Things like Time Magazine and the FDA or the what is it, Center for Disease Control or the CDC, those are generally pretty good sites. And so you can do those. But like I said, I've got in here on the references there that you should only cite papers scientifically appropriate from government information, USDA, FDA, CDC, etc., scientific journals, Nature, Science, etc., popular magazines that are known to be reliable, like Scientific American, National Geographic, Time, Newsweek, books and textbooks on biology, newspapers of a credible source like the New York Times. Do not give me something from the National Enquirer. A variety of scientifically reputable television shows like History Channel, PBS, where they try to be reliable and accurate. Sources to be suspicious, and you should not have as a reference include a religious, politically, or privately funded publication. And that is, they have their own viewpoint and they will only promote that viewpoint. Personal websites should not be used because anyone can say anything on the web and it may not be accurate. College and university pages that contain student work should not be used as a reference. You may use college and university pages from a professor because generally that would be reliable information. It's not always, but generally it will be reliable. And usually, like whenever I find a mistake in my page, I'll update it. Okay. Wikipedia cannot be used as a reference because anyone can go into Wikipedia and change it. I actually had a friend who went into Charles Darwin and he went in and put in a sentence just to test if it could be done. And we gathered that Charles Darwin would be something that they would keep an eye on because anyone can go in and change it. And he was able to make the change. The point is, anyone can put in anything at Wikipedia. And yes, it tends to be reliable, but you can't count on it because anyone can go in at any time and then change the information on Wikipedia's page. You may use Wikipedia, just don't give it as a reference. And then what you can do is give their references because their references are usually reliable sources. Is that clear? And where do I have that if there? Someplace I've got stated that the literature citations page you must have at least 4 unique sources referenced in your report that are reliable references. And I can just look at them and I will know if they're reliable references or not. Any question about any of that? So get your sources, get your information, and then write up your document. You should have a written report detailing the information in your writing. It must be typed, should be single spaced at 1-inch margin, and I do require 12-point font. Some of you have been turning in your lab work with very small print, and I'm having a hard time reading it even with my reading glasses. Okay? And that's going to annoy me. So I'm going to warn you. Usually I put it, don't use small font when you're turning in information to a professor, especially if the professor wears glasses. And by the way, these are not, what do you call the corrective glasses? These are reading glasses. All they do is they magnify. So when you're writing up your report, you should have your cover information. Do not have a cover page. Put all your cover information on the first page of your document. Your cover information should have your name, the title of your project, the date, the course name. Okay, that's about it. And then put in the text, the main body of your document on the first page. So that should be with your cover information. Don't have a cover page. If I were to print it out, that would just waste paper. You must describe all aspects of the disease or organism causing the disease and itemize them in the report content, which I'll talk about in a minute. The material presented in the text is worth 60 points. An additional 15 points are for good grammar, good spelling, good flow of the reading, and good readability. As much as possible, the text should be written in your own words. Use direct quotes only when absolutely necessary. I once had a student turn in a report where about 75% of the material was quoted. So she was not plagiarizing, but I told her that's too much. And now I will give you a penalty. I didn't give her a penalty because I didn't warn her in advance. If over 50% of the paper is quoted material, you will get a penalty. Okay. So you should only put what should be in quotes, what's absolutely necessary when you're quoting someone. Put it in quotes and say where you got it from. Otherwise, it's plagiarizing. And this paper will be checked for plagiarism. It'll be done by Turnitin if anyone's familiar with that outfit. Any information that is not common knowledge should be either directly quoted or paraphrased, and then after that knowledge, which is not common knowledge, you should put in an in-text citation where you got that information. And that should be after the information that you're stating. Let me go through here and give you an example. So let's say that you state something like you're doing your report on COVID-19 and you say something like COVID-19 has killed, I don't know what the numbers are, so we're just going to say over 3 million Americans. This is not common knowledge, so you should put in-text citation where you got that from. And that would be something like Smith, 2024. And that's how you do an in-text. You give the author, in parentheses, you give the author, comma, and then the date that you got it. If ever there's two authors, you go Smith and Jones, comma, and then the date. If ever there's more than two authors, you go Smith et al. 2024. And excuse me, that should be in a parentheses. I'm not going to talk about the other way of doing a in-text citation, but you could do something like, let's go right here. Smith in 2024 said, and generally most people don't use that. COVID-19 has killed over 3 million people. Mostly they'll say COVID-19 has killed over 3 million people, and then you give the in-text citation. So I'm just going to talk about it that way, because very few people do it the other way. All right. Any question about that? This et al. Is almost the only time you can use et al. In a, I don't know, a course publication. When you give the full references, all of the in-text citations, so this is an in-text, didn't do that right. Close enough. There we go. In-text citation. And then at the end of your paper, you should have the references. And by that, I mean the full references. you should have each in-text citation, but you have it in the long form. So you're going to go Smith, and then generally you just go initials. We're going to go AJ. And you could give out the first name, like Andy, and then give the year, and then the source where you got it from. Oops. Is there anything else in that I'm looking at? I'll talk about how you do the source in a moment. There's generally 3 ways to do a source. For a web, not a web, a scientific journal, you give the article name, the journal name, the volume number of the journal, and then the page number. So that's a scientific publication. Now, for a newspaper or for the internet, you would just do something similar. For article name, it would be like the name of the page. And then you give the web newspaper, which would be something like New York Times, or for the internet, give the web address. And it should be the exact web address so I could click on it and take a look at it if I wanted to. For a book, it's a little more difficult. And this you should have learned in the plagiarism quiz, by the way, or the tutorial. You give the article name, the book name, the book, so that's the author of the article, the book editor, assuming it has an editor. If the same author made the book, then you only need to give it one time. But if it's a different editor, you give the book editor. The book publisher, and the city of publication. For some reason, you don't give the country of publication. I don't know the reason for that, but that's just how it's done. So the author of the chapter or article in the book, the year, the article name, the book editor, the publisher, and the city of publication. Any questions about the full references and the in-text citation? All right. Oh, the point is, in the full references, you really should not use at all. There is one exception, and I even hate to state it, but if you have more than 8 authors, you have to give all 7 of the first authors. So we're going to go author 2, author 3, author 4, author 5, author 6, author 7, and then you can go at all. That's the only time you can use at all in the full references, when you have 7 or more authors. And that doesn't happen very often. Any question about that? And let me state that students frequently get the in-text citation or the full references or both wrong. And they'll miss a few points if they're doing something wrong. So if you're giving information that's not commonly known, you should give an in-text citation directly after that information. There is one exception, and that is if you have a whole paragraph where you're giving information from one author, you could then, instead of do it after each sentence, you could do it at the end of the paragraph. Is that clear? All right, the in-text citations are worth 10 points. You may use any commonly used citation format, APA, MLA, scientific format, anything you want. The only thing is be consistent. Don't use APA in one place and then MLA in another place. So if you use APA, use it throughout the document. And the in-text and the full references should be using the same format. And for the full citation, meaning the literature citation page, it should list all the sources cited within the paper, and then any that you use which you don't cite in the paper. That's the full citation. You must have at least 4 unique sources referenced in your report. The complete literature citation page, meaning the full references, is worth 15 points. The final report must be submitted to the folder on Canvas as an attachment, either as a doc, a docx, MS Word, a rich text format, or PDF format is no longer valid, sorry. Any question about that? I guess you could do a .text, but you won't have any formatting in a .text. Any question about that? It can't be PDF because the plagiarism checker struggles or cannot do a PDF format. So it has to be another format. If you don't have one of these possibilities, which everyone should have rich text format, just take your word processor and save it down as rich text format. And everyone should have text also. But you can go to a computer at Clark College to get a doc or docx format and submit it that way. The project context of the text, you should have each section below. Each section is worth about 10 points for a total of 60 points. Use these subject headings when you write your paper so that I know where each section begins and where each section ends. So you should discuss the causative agent. So you should have that in your paper. Causative agent is what's the causative agent of the disease? Is it a bacterium, a virus, a prion, or a eukaryote parasite? If it's a bacterium, you should discuss the characteristics of the cell. What's the gram stain? What's the cell shape? What's the cell arrangement? What are some metabolic capability, etc. If it's a virus, what are its characteristics? Is it a DNA virus? Is it an RNA virus? If it's an RNA virus, if it's single-stranded, is it positive single strand or negative single strand? Is it a retrovirus? Is it an envelope virus or is it a naked virus? If you don't understand this about the virus, we will talk about viruses later in the term. How large is the virus? Does the virus form a provirus? What are some unique characteristics of the viral multiplication cycle? And you could state that for any of the infective agent. What are some unique characteristics of this disease? If it's a prion, tell us what is a prion? Where in the body does it occur? What's the function of the normal type of protein? A prion, if you don't know, is caused by a protein. If it's a eukaryote, state whether it's a fungus, an algae, a protozoan, a platyhelminth, or a nematode. Is it multicellular or unicellular? And then give its life cycle. The next section is history. So your paper should have a subheading history. How long have we known about this disease? Historically, how was this disease treated? What is the history of the vaccine development for this disease? If the disease has no vaccine and there's no vaccine, what's the word, in research, state that. Most diseases either have a vaccine or there is a vaccine that they're developing, meaning it's in research stage. Like AIDS has no current vaccine, but they've actually made a few different vaccines, and obviously none of them have worked satisfactory, or there's other problems with the vaccine, so it's never been used. Describe the changes in our knowledge and attitudes towards this disease throughout history. Even for COVID-19, which is a really recent disease, we've changed our knowledge greatly and our attitude greatly about this disease. So talk about that. And that should be under history. And then talk about epidemiology. Describe the prevalence and transmission of the disease. How is it transmitted from patient to patient? Where in the world is the disease prevalent? How many people are currently infected? What is the rate of new infections per year? What's the rate of morbidity and mortality of this disease? Some people don't give that, and for something that is very mild, like the chicken pox, you should still give the morbidity. The mortality rate for the chicken pox would be very, very low. You could give that, like each year there might be 2 to 5 people dying in the United States from the chicken pox, but you can always state a little bit about the mortality, the morbidity. So you should state that even if it's very low. What is the reservoir of the pathogen? Like malaria, everybody knows it comes from obviously people or mosquitoes. What is the mode of transmission? How does the patient pick it up and then get it? Pathology. Describe the pathogenic effects on cells, tissues, and organ systems. What tissues, organs, cells does the pathogen infect? What damage does the pathogen inflict? How is the damage inflicted? Is there direct mechanical damage by the pathogen, or is it a toxin produced by the pathogen? Or does the immune system respond and cause the damage? What is the time sequence of the disease? The length of incubation? What's the prodrome period, the illness period, the decline period, the period of convalescence? What are the major signs and symptoms of the disease? Now, let me state that occasionally we have a disease where the damage is not known. Like this came as a surprise to me, but I did research on it, that they do not know why. Let me see if I can remember the disease now. Why rabies tends to kill people. They do not know what the damage is that rabies does. Okay, so you need to state that. Do your research. If you can't find anything, try and find a statement that tells you that the exact damage that this disease is doing is not understood, and then give that as a citation. Most diseases, they do know what the damage is. What are the major signs and symptoms of the disease? And that's usually an easy one. And then the response and treatment. Describe the activity of our immune system against the pathogen. Is it cell-mediated immunity, humoral immunity? That's with antibodies. Is it innate immunity? That would be something like a child getting a cold for the first time. They have innate immunity to getting the disease. And by the way, that's why children are much resistant to COVID-19 than elderly people. Children have a good innate immunity, and that tends to hit COVID-19. And then is it acquired immunity? That's when your immune system learns to respond to a specific pathogen. So initially you don't respond, but as the pathogen resides in you for a week, you start responding. In about 2 weeks, your T cells, it's a immune system cell that responds, and then your antibodies start being produced to attack the pathogen. How is the disease diagnosed? What types of medical treatments exist? Describe how these treatments affect the progression of disease, antibiotic resistant strains, etc. Describe prophylactic measures that can be taken to limit the risk of an infection. Like you avoid people who are sick. That's definitely one thing you can do. And then talk about the socio, political, and economic considerations of this disease. I'll let you read about that. I have a grading rubric found on the next page. This is how your assignment will be graded. You'll get 2 points for the cover information. You'll get 9 points for the causative agent, 10 points for the history, 10 points for the epidemiology, 10 points for the pathology, 10 points for the response and treatment, 9 points for the social and political and economic issues, 15 points for your quality of writing, 10 points for your in-text citations, 15 points for your literature citation page, meaning your full references, for a total of 100 points. Any question about any of that? Let me see if there's anything else I need to talk about. I don't think so. So let's go take a look at the web page. Let me see if I've got a there's the infectious disease project. If I don't have an example, I'll try and get an example up here. I think that was new, so I probably don't have it in the summer. Yep. All right. I think that's it. I'll make a note that I put the example. You got the grading already. It should be in with the infectious disease project. Oh, there it is right there, the infectious disease example. I somehow missed it. All right, any questions about the infectious disease project? If not, let's go on to lab module 2 and the worksheet. All right, lab module 2 is on aseptic transfers, the learning objectives. Upon successful completion of the lab, you should be able to understand the purpose of using aseptic transfer techniques. 2, determine if aseptic transfer is used from the data provided. And then 3, define the following terms. What's ubiquitous, aseptic transfer, inoculation, and a contaminant. In our local environment, microorganisms are ubiquitous. This is problematic when working in a microbiology lab because we generally want to work with a pure culture, a culture that contains one and only one species of organism. Louis Pasteur developed the technique of using aseptic transfers to minimize the risk of culture contamination. He also came up with a method of getting a pure culture, but we don't use his method anymore because he used limiting dilution of a liquid solution, making many different dilutions and then replica plating those dilutions. And it was a difficult way of getting a pure culture. We will talk later about how we get a pure culture. And then in today's lab, we'll learn how to make an aseptic transfer. Some important things with aseptic technique. During inoculation, never lay the test tube cap or petri dish lid on the workbench because they are not sterile and they will pick up organisms. And then when you take the lid or the lid or the cap, and then bring it over the tube or the dish, something could fall down onto the dish and inside the dish. To minimize contamination when inoculating media, tilt the tube at an angle. Let me see if I can show this. I don't have a good thing. I just use my cup. I got water in here, so I can't tilt it very far, but you should tilt it at an angle, and that's not very good. So let me try and do it this way. So that when you are working inside, instead of having the full surface where something can fall down into it, you only have a little bit of the surface that something can fall down and go into it. And then when you're working with the petri dish, you should take the lid and cover the dish. I don't know if you can see this or not, but I'll try and do it. Take this out of here. Cover the dish, which you guys will be on a table, so it'd be a little easier. And then have the lid covering the dish so that most of the dish is protected. Only this area right here is exposed to something falling down from the air. And then work quickly so that you can then cover the dish as soon as you can. Any questions about any of that? Work efficiently and quickly. Obviously, you shouldn't talk while you're doing it. Shouldn't be walking around while you're doing it, things like that. In order to inoculate, that's transfer cells to a new sterile media, we must make sure that we sterilize everything that can potentially contaminate the media. And how we do that is we use a Bacto incinerator, but you could use a Bunsen burner and a wire loop or a tool, something like this. You pick up your bacteria with the wire loop, but before you do that, you put it in the Bacto incinerator or put it into the flame, and you get this wire portion, the metal portion of the loop, red hot to kill anything that might be on it. You do not make the metal portion of the handle red hot, and obviously the plastic or rubber you don't put in the flame because you just melt it. Any question about that? You then let this cool. You could let it sit in the air for 20 or 30 seconds to let it cool, meaning hold it. Or more likely, what you do is you take it on the tube, touch it to inside of the tube, which is sterile, to cool down the loop, then take the loop out of this tube to pick up the cells in the stock culture, and then move them into your sterile media. Any question about any of that? Know the terms of this lab? You're not physically going to be performing any of these activities, but you will be watching videos of these activities, and we expect that you will know and understand the material in these videos and in this lab module. In procedure 1, we're going to transfer sterile liquid using a glass serological pipette. I'm going to skip right here and I'll come back to procedure 2. We have a little video for you to watch on demonstrating proper aseptic techniques. So watch that video. And in procedure 1, you're going to transfer 1 ml of water, sterile water, in a sterile glass tube into a tube containing sterile media. And it's problematic when you measure the water, especially in a glass tube or a birette, because the water does not form a flat surface in the pipette or the birette. It forms a meniscus. And how you read the meniscus is you read it from the bottom of the meniscus. You don't read it from the top of the meniscus. You don't read it from the average or the middle of the meniscus. You read it from the bottom of the meniscus. So in procedure 1, you take 1 ml of sterile water, put it into a sterile test tube. And then you incubate that tube. And there we have the tube of media before use. It looks here. Broth cultures are problematic when we're using them, and we're going to use a broth culture for procedure 1, because if you have minor contamination, you will not be able to tell it from the major cells, meaning most of the cells are the cells that you inoculated, because all the cells are going to mix together in the broth. If you have major contamination in the tube, you probably will be able to tell it, because the media could take a different color or could look wrong. But minor contamination, you would not notice it. And then use this table for making your observations and conclusions. And then when you're writing up the answers, your result and the conclusion should follow wording similar to this table. For procedure 1, if the nutrient broth is clear, the conclusion is no organisms are growing in the broth and aseptic transfer was successful. For procedure 1, if the nutrient broth is turbid, meaning hazy or cloudy, the conclusion is organisms are growing in the broth and aseptic transfer was unsuccessful. Any question about any of that? As I stated, your wording must be similar in the results and the conclusions from this table. It doesn't have to be the same, although it can be the same, but you must use wording similar to the table. For procedure 1, here is the tube after you've taken it out of the incubator for P1, and then answer the questions for 1. First, describe, and you'll have this as a 0, describe the initial clarity of the nutrient broth prior to inoculating, meaning prior to adding sterile water or the paper disc for procedure 2. Was it clear or turbid? So your answer is going to be clear or turbid. Okay? And then in procedure 1, describe the clarity of the nutrient broth after inoculating with sterile water and overnight incubation at 35 degrees Celsius. Was it clear or turbid? Based on these results, what conclusion can be drawn? Use the table. And then that'd be 1A, 1B, this would be 1C. 1C, list 3 sources of error with aseptic transfer technique that could explain why the nutrient broth showed growth of microbes. So give me 3 possibilities. And if it's possible, I will accept it. If it's not possible, I won't accept it. Any question about that? All right, that's it for procedure 1. Let's move to procedure 2. Procedure 2, you're going to transfer a sterile paper disc using clean forceps. You can also watch the video there, but you only need to watch it 1 time. So using aseptic technique, transfer a sterile paper disc into a nutrient broth, and here's the procedure here. You can read through it. I guess with this one, I should sort of state it. And you're going to use forceps, which are not sterile. And I don't have forceps, but I got scissors. We're going to pretend these are giant forceps. And you're going to come into the sterile paper disc. We're going to pretend this is a paper disc. And to clean the forceps, you dip them in alcohol and then you flame it. Either put it through the Bunsen burner or put it inside the Becton incinerator so it catches fire. Let the fire burn down and then pick up the forceps and put it into your glass tube. Okay, pretend that's an angle, stick it in. Any questions about that? Once again, there's the tube you're sticking the paper in. Use the table for procedure 2. If the nutrient broth is clear, the conclusion is no organism are growing in the broth and aseptic transfer was successful. If the nutrient broth is turbid, hazy or cloudy, organisms are growing in the broth and aseptic transfer was unsuccessful. There's tube P2 for procedure 2. You can actually see the paper disc down at the bottom of it. And then answer questions 2. For results P2, describe the clarity of the nutrient broth after inoculating with a sterile paper disc and overnight incubation at 35 degrees. Was it clear or turbid? Based on these results, what conclusion can be drawn? Any questions? All right, for procedure 3, you're transferring Serratia marcescens from a stock culture to a broth culture. We have a little video for you to watch on that. This video does not use Serratia marcescens. It's using E. coli. Serratia marcescens is a little easier to work with because its color is red. E. coli is off-white. You only need to watch the video to 4 minutes and 23 seconds. There's an error that's made in the video when the instructor puts the loop into the E. coli stock culture tube. You can see it someplace between 3 minutes and 5 seconds to 3 minutes and 20 seconds. Do you know what that error is? You can hint, you can hear sizzling when he puts the loop in the E. coli, and you can just state what he did wrong. In procedure 3, using aseptic technique, transfer Serratia marcescens to a sterile broth tube. Serratia marcescens will grow, excuse me, pink when it's grown in a liquid culture. And it's true your tube will look like that to begin with, but that's not a question. Use your table here for procedure 3. If the nutrient broth is clear, the conclusion is no organisms are growing in the broth and aseptic transfer was unsuccessful. If the nutrient broth is turbid, hazy, or cloudy, organisms are growing in the broth and aseptic transfer was successful. Why, in procedure 3, is the transfer successful if the nutrient broth is turbid, but for procedure 2, if the nutrient broth is turbid, the aseptic transfer was unsuccessful? Can anyone explain that? All right, think about it if you don't know. There's the results of procedure 3. It's a broth tube after incubating in the incubator for overnight at 30 degrees, and state whether the clarity of the nutrient broth is clear or turbid. Based on the results, what conclusion can be drawn? And how do you know whether you transferred the correct microbe, Serratia marcescens? All right, for procedure 4, we're going to be once again transferring Serratia marcescens, but not to a liquid broth. We're going to be transferring it to an agar solid slant. Serratia marcescens, when it grows on a solid slant, will have a red pigmentation to its color. Contamination can be more easily detected on a slant than in a broth, because even if you had minor contamination, like right here, it will be a different color right there where you have the contaminant than everywhere else on where you're streaking it. Is that clear? So even with minor contamination on a slant, you can determine the contamination. In a broth, you cannot determine if there's been minor contamination. All right, for procedure 4, use this table. If the slant does not show confluent or colony growth, no organisms are growing on the slant, and aseptic transfer was unsuccessful. If, on the other hand, the slant shows confluent or colony growth, the conclusion is organisms are growing on the slant and aseptic transfer was successful. All right, P4 is the slant after we've grown the tube in the incubator overnight at 30 degrees Celsius. For procedure 4, is there bacterial growth on the slant? You just need to answer yes or no. How do you know? Just state that. And does it appear to be bacterium inoculated or a contaminant? And then explain why. Any questions about this lab? If not, get to it. I'll be here until 9 o'clock, or the last student logs out, to help you with any questions. All right, I'm going to stop the share and stop the video.